And thank you all for joining us today for this talk. Here are my disclosures. I have nothing to disclose here. And so the objectives for this talk will be to understand the importance of rapid next generation sequencing based approaches to detect and identify microbes in the laboratory. To also learn how custom ion AmpliSeq targeted NGS panels can be used alongside with the Ion Torrent Genexus integrated sequencer to deliver NGS tests within as little as 24 hours. And then lastly, to understand the performance of the new rapid microbial identification method and how it has the potential to be incorporated into standard microbiology testing. I'm going to start the portion of my talk just focusing on the clinical need for this application. Microbiology has evolved significantly over the past couple of decades in particular. And the infectious disease approach of culture has progressed and has been supplemented with molecular testing, either directly from isolates or from specimens. And in the recent years, this has progressed further into sequencing options, including Sanger sequencing, but also more interestingly, at least in my opinion, the next generation sequencing. So ultimately, as a laboratorian, we're always looking for that unicorn when it comes to diagnostic tests. We want it to be good, we want it to be fast, and we want it to be cheap. However, this can be quite difficult to really obtain in the diagnostic setting. And so typically, you would, if anything, accept 2 out of 3, which isn't so bad. Currently, culture is cheap and good, but not necessarily very fast. Antigen testing is cheap and fast, but not necessarily the greatest when it comes to performance. And PCR may be are considered good, somewhat fast depending on the methodology. And so the question really is, where does NGS fit into this box here or these circles here? It's certainly good, but how do we address the other factors into making it fast, in particular, which is an area that that I want to focus on. So in 2021, I was fortunate enough to be involved in the development of this consensus study with the National Academy of Science. And our focus was to combat antimicrobial resistance and the protection of the modern medicine. Um, in the document, we included this figure, which really identified factors that contributed to antimicrobial resistance. And not surprisingly, one of it is just the overall misuse and overuse of antimicrobials. And the second part that I find also very relevant is the fact that there really is suboptimal rapid diagnostics available right now, and this does contribute and has been found to contribute to the overall increase in antimicrobial resistance. And the pandemic certainly contributed significantly to this as well. And in our report, we commented on how there were considerable presumptive antimicrobial use during the pandemic in patients that were diagnosed with COVID-19. And this was a very much of a just in case approach in prescribing. There was one study that was published in 2020 that reported that despite the fact that only 7% of COVID-19 patients had secondary bacterial infections, over 70% of which were treated with antibiotics. And then another study in India found that there was an excess of over 200 million doses of antimicrobials that were used during the pandemic. The report that we compiled worked off of the previous 5-year action plan and built on it and I wanted to focus on the 2 points that's in the pink box right there. One is to support innovation in developing and deploying diagnostic tools that would improve the overall diagnosis and treatment of the patients. And then lastly, to also be able to collect and compile data to have us better understand what diagnostic tools would be an appropriate option to meet this need. So there certainly has been an increased interest and even a little bit of FOMO when it comes to the utility of next generation sequencing for infectious diseases. And even though it's this bright and shiny new tool, it really does have to have the same approach when determining or not to use it for clinical diagnosis. And the benchmarks that applies to this is whether the result that would be reported would alter a patient's management and affect outcomes. Also, the emphasis on turnaround time is key to allow for timely patient management because if you're on empiric therapy for a prolonged period of time and the results come out even longer than that, there's very minimal impact that can happen. And then lastly, to be able to be used as a tool that it can potentially avert any other testing that may be deemed unnecessarily unnecessary. And this, if anything, can help contribute to offset some of the costs of this molecular test. So because of this interest and my curiosity in the past, about 3 or so years ago, I did send out a survey through the clinical microbiology listservs. This is through the American Society for Microbiology. And the question that I asked the laboratory directors was, does your clinical lab offer infectious disease NGS as a clinical test? And of the 104 respondents, there were only a total of 19 sites that reported out as yes, they did perform this in-house. And when you separated out the clinical labs, there was only 16% of those labs that offered it in-house. And not surprisingly for public health, there was 50%. So despite the fact that with the bulk of the labs saying no, my next question to them was, is this being sent out to reference labs? And the vast majority, more than 80% of which determined that answered that it was being sent out. So some key takeaways from this survey was that there was only a small percentage of clinical labs that are offering NGS testing in-house right now for infectious disease testing. But however, there definitely is an interest and definitely a need as more than 80% of these labs are sending it out to reference lab for testing. And if anything, this offers us opportunities to improve and to grow and to really answer the questions as how we could potentially bring this back in-house. The next generation sequencing has a number of different applications. One is whole genome sequencing for species identification directly from specimen or from isolates, which allows you to also determine resistance mechanisms, outbreak investigations, etc. The next application is targeted next generation sequencing, mainly for the identification to the genus and species level of a variety of different organisms, depending on the target that you have included. And then lastly is metagenomics NGS, which is the unbiased approach, shotgun approach of detection of pathogens, where you can also detect parasites, etc. And so for the purpose of this talk, I will be focusing on targeted NGS because that was the approach that we pursued for our laboratory. And so one of the primary reasons why we looked into this was that there certainly, as I mentioned, there certainly was a need, and we were seeing this even in our laboratory where there was an increase in volume over time. And if you look at even in recent years, the increase was quite dramatic. And then, of course, it comes with an associated cost that we in the department would be having to take on. And so you can see even from FY22 or 22 to 23, is just a huge expansion in the interest and the orders of these tests. Looking back at some studies and the overall impact that these types of targeted 16S or other targeted assays had on overall patient management. There was one paper that was published in 2020 that's listed here that looked at, I believe, 607 clinical isolates and where they offered culture, bacterial 16S ribosomal RNA, PCR and sequencing, and then targeted PCR on these samples. One thing I wanted to just note about what they found in the study was that the specimen choice that offered the highest yield was pus samples, abscesses, etc., whereas the lowest yield was found to be CSF. And another study corroborated these findings, where they also reported lowest yield to be CSF and blood samples. The interesting thing about the study was that it found that 16S ribosomal RNA, PCR and sequencing de- detected additional organisms at set 47 compared to what was identified by culture and PCR. And so some of the takeaways from that paper that I summarized here includes the relatively low overall positivity rate of 12.9% that were positive. And I'll touch on that a little bit more later with some in the coming slides. But what also was reported was that 53.2% of the positives that occurred were new diagnoses that were solely made by the 16S ribosomal RNA. And of the 91 patients that had prescribing information, there was a change of antimicrobial change that occurred in 14.1% of these patients. So relatively low. And in the cases that were culture negative and 16S rRNA positive, the mean cost that they guesstimated or calculated that a 16S positive culture negative result uh, equated to about £4,000 for one prescription change. So because of the low yield of positivity and the cost of the test, that was the calculation, was that is about $4,000 a pop for any positive result that they identified. And one thing I wanted to highlight from that study was that the mean turnaround time was 13 days, which is significantly longer than what they would have preferred and what we would expect. And a quote that I included from the authors states that laboratories with core sequencing facilities typically have turnaround time of 16S PCR and targeted PCR of 24 to 48 hours, which reflects the potential impact of repatriation. So basically, being able to bring these types of tests, these molecular tests in-house, may allow them to shorten the turnaround time. And there was another study that looked at a retrospective observation of 310 samples in 268 patients, looking at targeted 16S and 18S rRNA testing. And overall, the positivity for just the molecular test itself was low. But same thing, the impact was also very minimal. There was only a change in antimicrobials in 6 patients, so 2.2%. And for these targeted fungal tests, there were no changes in antifungal therapy. They reported a turnaround time of about 7 days, which is still faster than the previous study, but they did also mention that they felt that the minimal impact on patients was attributed to the fact that the turnaround time was delayed. We did a similar study about 6 years ago, summarizing 78 samples that were sent for 45 patients to a reference lab for targeted 16S and 18S or ITS sequencing, PCR and sequencing. And we found very similar findings, 14% positivity rate, which is quite low. We found that there were a useful diagnostic input in 7 of these cases. However, there were only antimicrobial change that was made in 1 patient. So overall, low yield as well. Some considerations from our study, overall, there was a low impact on patient management, and we attribute it also to the fact that the turnaround time was 11 and a half days, and even up to 14 days in some cases. And we commented on the lower positivity rate of 14%. And, some postulations was that, is it because of overtesting? Are we including specimens or cases where it likely is a non-infectious etiology. The probability of a test actually picking up a pathogen is quite low, which then dilutes, the true positive cases. And also, at the time, we're sending remnant samples in many cases that perhaps would have sat around in the refrigerator for a bit before it was sent out. So that could potentially have also contributed. So overall, there are factors that affect clinical utility of NGS. Case selection issue, the right population, as I mentioned, um, whether you would be sending it out after conventional microbiology results is available, treatment therapy and treatment failure potentially being a factor as to why you may send. Sample collection issues as well, the timing of the collection, the sample type. For example, sending out a swab for NGS is likely not optimal. Result interpretation as well. This is important because it is a more sensitive test. You may be picking up contaminants, dead microbes, etc., that may be considered a clinical false positive, but also clinical false negative in some cases. And then lastly, of course, the issue of expenses. It's not covered any reimbursement by insurance currently. And then, as I mentioned in some of the other slides, just the importance of the turnaround time. And I do think that there are improvements and opportunities for improvements. Of all of these factors, if a test can be brought in-house, it wouldn't necessarily address all the issues, but at least there may be more oversight with implementation of diagnostic stewardship efforts, um, the utilization of the medical microbiologist to help with the interpretation, using in the context of the clinical presentation, which may not necessarily be available to the reference laboratory that are offering these tests. So what would be optimal? So as a lab director, what would I want to see when it comes to NGS and how I would be able to bring it into my lab? So optimally, I want it to be as close as possible to the clinical laboratory. And at this, using that term, I'm calling it the point of care. Um, but this is, of course, easier said than done. There are multiple hurdles, as would be expected for any lab test, but more so for a highly complex molecular assay. So, what are the problems that would need to be tackled for us to be able to even consider bringing this on into the clinical laboratory? And there are many problems, and I've only listed a few here. I'm sure there are many more, but ultimately, the infrastructure. Does the clinical lab even have an infrastructure for pre-analytical, analytical, molecular setup? Molecular expertise, of course, is going to be extremely important. These are highly complex procedures that are involved, and it also requires extensive hands-on time to set up the library preps, etc. Clinical microbiology tends to not have a lot of expertise when it comes to bioinformatics. And so having access to that to help with the post-analytical process and to simplifying it so that it can be reported out in as streamlined a process as possible. And then, of course, as I mentioned before, the cost and the reimbursement is definitely an issue. And this slide just really illustrates the complexity of the different steps that are involved for any NGS application, whether it be whole genome sequencing, targeted sequencing, or metagenomic. And you may incorporate some automation for pipetting, but ultimately it still is a very manual process. And in order for it to be realistic or feasible to be brought into a clinical microbiology lab, a lot of these steps would have to be eliminated. So where we need to go from the specimen, pop into the machine with minimal hands-on time and preparation of the specimen. Plug and play is really the key. We want it to be at a state where you can have small batches of samples that are tested, which will also improve the turnaround time. You don't want to necessarily, depending on the lab, but certainly from my lab, you don't want to necessarily have to wait for 96 samples to come in before you pop it into the machine either. So being able to have like those smaller sample volumes would be very helpful. And then for same thing from the informatics side, having a plug and play approach that would spit out the results where we can get the identification, depending on the approach, the potentially resistance profile, etc. That would be extremely helpful. And tying it back to stewardship, which is a huge passion of mine. Really, ultimately, being able to bring it in house, allow us to have like that direct oversight, where we allow for the right test for the right patient and at the right time. And this is, the question for diagnostic stewardship in many cases is whether it's to test or not to test. And the microbiology laboratory play a key role in both diagnostic stewardship, but also in antimicrobial stewardship, which really all ties together. And I would argue that from the diagnostic stewardship point, having that microbiology expert, that medical microbiologist be that key person or help lead that committee to guide diagnostic testing selections would be extremely helpful for the institution. So you really want to find that sweet spot. You don't necessarily want everybody to get that test where it's a free for all, because of course there are considerations, including a negative patient impact. But you also don't want to be that grim reaper that does not allow any testing at all. So, wanting to kind of understand what that perfect sweet spot would be. Um, so with that, when you're taking into consider what that spot would be, you want to maximize impact. And with that, it really is to have judicial use of only patients that have high suspicion of infection. And you can define this in multiple ways, perhaps in specimens with higher yield, such as organisms seen on a gram stain, but it's negative by culture. And then, of course, emphasizing that collaboration, because that is extremely important, and it doesn't help anybody to be siloed into your own sections only. And so the NGS playbook, as I put it into 4 rules, would be, to have an approval process for this, whether it be offered in-house or outside as a reference lab, restrict or testing on multiple specimens, and to reserve for difficult cases where perhaps standard of care testing have been negative, or critical cases where will directly quickly have an impact on that patient. And then to emphasize that this is certainly not a rule out test in any way, and you really do have to correlate it with clinical findings and other ancillary testing. And tying in the results that will be coming out, it's just emphasizing that these are complex tests that certainly do need some aid in interpretation, and to utilize your laboratory experts, medical microbiologists that can really help with this. And so myself and Dr. Christina Castellas, who's a colleague of mine here at CHLA, worked on this review article where we, talked about potentially modeling it after oncology with their tumor board discussions. Perhaps we call it an abscess board, nodule board, pus board, whatever it is. But, it's to have like that direct communication with physician. Avoid having our ID colleagues who we work really well with, but having to have them be that middleman so that you can directly, explain your reasonings behind certain things, and also the nuances of certain diagnostic tests and the interpretation of it. So collaborating in that way, discussing the sequence results, determining perhaps whether except modifications of cutoffs can be had depending on the case, like all these things that could be very helpful in a case by case basis. And now we're going to shift to the feasibility of a new targeted metagenomic NGS test for rapid microbial detection and identification. I'd like to present the Ion Torrent Genexus system, which is an image of it here. This system offers an end-to-end workflow from the biological sample to the report. It is quite fast as 24-hour turnaround time, which allows you to provide those results. It is automated in the sense that there's a sample extraction, quantitation, library prep, sequencing, and analysis process. It also allows for the reduction in potential manual errors that would be had if it was a person that's going to have to do all of the pipetting, etc. And it also has the flexibility of economically running as few as one sample, reducing the need for batching, which is something that I mentioned earlier is a need, particularly for labs that are much smaller in size. The system itself and the consumables are manufactured at facilities registered with the FDA and ISO. It's 13485 certified. And the research applications, although, it's not as interesting as infectious diseases, just kidding, but there are other applications such as oncology, reproductive health, and inherited diseases. So we had the pleasure of using the GenX's system during the pandemic, where we were performing whole genome sequencing for SARS-CoV-2 for positive samples. And one of the reasons why the instrument was of interest to me was the automation capability, the minimal wet lab hands-on time, as depicted by this table that we published in JCM in 2022. It's 10 to 20 minutes of wet lab time versus we calculated about 245 minutes with the Illumina that we were also using at the time. And the turnaround time, because you can run smaller samples and because it is so automated, we were able to complete one sample within 48 hours from the time the specimen was received in the lab, and which would include having to perform the PCR to confirm that it is positive for SARS-CoV-2. So it was a significant game changer for us. So the next steps, after the pandemic, the approach that we wanted to look into was targeted NGS for bacterial, fungal, microbacterial. We wanted same thing to prioritize that automation that the GenXus is capable of, which would allow for hopefully reduced costs when you think of labor costs and then reduced turnaround time. So we named this whole study Amplified for automated targeted metagenomic next generation sequencing pipeline for rapid infectious disease diagnosis. And the entire study was performed on the Ion Torrent GenXus system. And collaborating with the Thermo Fisher team, we worked on designing a panel that targeted both bacteria, including microbacteria and fungus. And this included 16S gene, but also species-specific gene that was developed by the Thermo Fisher scientific team, and then the ROBI for microbacteria. We also wanted to include fungi using the ITS and D1, D2 region. And our focus was to ultimately include specimens that we are currently sending out. So that included sterile body fluids, tissue, and then we wanted to also include isolates. Okay, so the workflow that we established is a automated DNA extraction system. And following that, and that takes about 15 minutes of hands-on time. Following that, we then had about 30 minutes of hands-on time to prepare the specimens and to load the reagents onto the GenXus instrument itself. Once that happens, the rest of it is all internal, where there are automated amplification, library prep, and then sequencing all within the machine. So by this point in time, our staff could walk away. And then after that, there is the FASTQ demultiplexing adapter, so that occurs within the GenXus itself. We then incorporated a CLC genomic system using from QIAGEN for the read filtering and clustering. And then we also, working with our informatics group, developed some tools to help streamline just the the interpretation and analysis of the data itself. And so basically, what our very talented bioinformatician developed was this dashboard. And this is a snapshot which illustrates a very user-friendly interface for filtering and also prioritizing species identification that would deem to be true positives. And the species that were listed are on the left-hand side of the column, and the dots that are present here are basically indicated by, size and, color. And so relative size of the dots correlate with the relative abundance of organism that was present, and then the color correlates with the specific reference database that was used, to make this interpretation. And then for the study, within the study itself, we included 50 samples that were tested on the AmpliSeq panel. And looking at the retrospective data of these patients, we classified them as either not infected or infected. So there were 16 cases that we deemed to be in the absence of infection due to the fact that they did not have an ICD-10 code that was infection related, other inflammatory markers, fever, etc. And then we had 34 patients that we deemed to be true infection, same thing based on some of those parameters. And this is a breakdown of the specimen type, with the majority of which being sterile fluids, followed by tissue. And then breaking it down further, we compared it against conventional culture. So and I'll have another slide that will summarize this, but for this particular slide, I wanted to focus on the last portion, which is the concordance between infection status and the metagenomic NGS test that we used. There were a total of 17 that were culture positive and metagenomic NGS negative, and then there were a total of 30 that were culture positive and also NGS positive. Focusing on the ones that were that were discordant, um, the 7 that were culture positive but NGS negative were actually found to be common contaminants and insignificant. So that was great. So if anything, NGS correlated more with clinical findings than the culture did. For the 10 that were true infections, but NGS missed, the majority of them were very weak positive with like one colony that grew negative gram stains. But there also were about 2 or 3 that were just below the threshold. So it was detected, but just below the threshold. And this would be a prime example of where you have that flexibility to look further into the data to determine how you might potentially modify that threshold. And then this table here, looking at the overall characteristics. So of the 50, as I mentioned, 34 were considered to be infection, 16 were not. The overall clinical sensitivity based off of these findings for the NGS assay was 54.6%, and specificity was 85.7%, which actually correlated quite well with with other findings in the literature. And the NPV and PPV were in the 70s. And then when I looked at the analytical performance itself, the sensitivity, when you're comparing it just straight to culture, was 79.2%, and specificity was at 100%. And this is just an example illustrating just we sequenced one tissue. It was an explant RVPA conduit that we tested, and you can see just the abundance of the lactobacillus that was detected by NGS compared to the negative control. You can just see a bunch of random. So overall, automated, targeted genomic NGS directly from specimens. Um, we the greatest thing about it with regards to the GenNexus system is that lab in a box approach where from the specimen, everything that is illustrated in this middle picture here is all done within the instrument itself, and it allows so that reduced turnaround time of even about 10 to 2 days, as I estimate, but even up to 24 hours, depending on the timing that it's set up. The AmpliSeq targeted NGS panel, it does offer on the Ion Torrent GenX's integrated sequencer, it does offer a quite a promising approach to to pathogen detection and to identification. So in summary, NGS has the potential to be an integral part of the clinical laboratory for pathogen detection. To maximize the impact of overall NGS, though, there really is the need to shorten that turnaround time. And the automation of NGS assays will potentially allow for that broader adoption in the clinical setting so that it's not reserved for the larger labs that currently have the infrastructure to perform these high complexity testing. And with that, I'd like to just list off some acknowledgements here that have really helped us this far with the study, particularly um, my lab staff, but also the staff in the clinical microbiology. A shout out to Dr. Lucas Osborne, who was the clinical fellow that worked on this study and really led and made the study happen. And then from the informatics side, Dr. Hakim Javadi, who really worked and developed this great bioinformatic tools that we can use to pair it with the GenX's system. So with that, I thank you for listening and would be happy to answer any questions later.